Research Work 2. Title: CRISPR-Cas9 Gene Editing, a revolutionary tool for precision genome engineering. Abstract: this research work provides an overview of CRISPR-Cas9 gene editing technology, its underlying mechanisms, and its applications in precision genome engineering. The paper explores the significance of CRISPR-Cas9, discusses its advantages over previous gene editing tools, and delves into its potential implications in various fields such as biomedicine, agriculture, and synthetic biology. It also highlights the challenges and ethical considerations associated with CRISPR-Cas9, as well as future prospects and directions for research in this rapidly evolving field. So, what is CRISPR? Gene editing. CRISPR gene editing refers to a revolutionary technology that allows scientists to make precise changes to the DNA of living organisms. CRISPR stands for Clustered Regularly Interspaced Short Palindromic Repeats, which are segments of DNA that are derived from bacteria and act as a defense mechanism against viral infections. The CRISPR-Cas system, which includes an enzyme called Cas9, is utilized to target specific sections of DNA and introduce modifications with high accuracy and efficiency. By using a guide RNA (guRNA) that is complementary to the target DNA sequence, the Cas9 enzyme can bind to and cut the DNA at the desired location. This cut can then be repaired by the cell's natural DNA repair mechanisms, resulting in the addition, deletion, or modification of genetic material. CRISPR gene editing has revolutionized the field of genetic research, enabling scientists to study gene functions, develop disease models, and potentially treat genetic disorders in the future. Background and significance of CRISPR gene editing. CRISPR gene editing has emerged as a revolutionary technology in the field of precision genome engineering. Building upon the natural defense mechanism found in bacteria, the CRISPR-Cas system allows scientists to precisely modify genetic material with unprecedented ease and efficiency. This breakthrough has transformed biomedical research, enabling targeted gene modifications, disease modeling, and the potential for innovative therapies. CRISPR gene editing holds immense significance as it offers a versatile and cost-effective approach compared to previous gene editing tools. Its impact extends beyond the laboratory, holding promise for advancements in medicine, agriculture, and biotechnology, with implications that will shape the future of science and healthcare. The need for precise and efficient genome engineering tools arises from the complexity and intricacy of the genetic material in living organisms. Genomes consist of vast amounts of genetic information that determine an organism's traits, functions, and susceptibility to diseases. To understand the precise functions of genes and their role in various biological processes, scientists require tools that can accurately modify specific regions of the genome. Additionally, the ability to precisely engineer genomes is crucial for applications such as developing targeted therapies, creating disease models, improving crop traits, and advancing biotechnology. Precise and efficient genome engineering tools, like CRISPR gene editing, enable researchers to manipulate genetic material with high specificity, allowing for greater precision in scientific investigations and practical applications. The historical context of gene editing technologies reveals a progression of scientific advancements in the field of genetics and molecular biology. It began with early studies on genetic inheritance by pioneers such as Gregor Mendel and the discovery of the structure of DNA by Watson and Crick. The advent of recombinant DNA technology in the 1970s enabled the insertion of foreign DNA into organisms, marking the birth of genetic engineering.
TLC-EDU animated board for detailed discussion.
The emergence of gene editing tools like zinc finger nucleases (ZFNs) and transcription activator-like effector nucleases (TALENs) paved the way for more precise modifications. Finally, the development of CRISPR-Cas9 gene editing in the 2010s brought unprecedented efficiency, simplicity, and versatility to the field, revolutionizing genome editing and opening up new frontiers in biology and medicine. The historical evolution of gene editing technologies reflects the continuous pursuit of understanding and manipulating the building blocks of life. Previous gene editing tools had several limitations that hindered their widespread adoption and application. Some key limitations include: 1. Complexity and limited targeting. Earlier techniques like zinc finger nucleases (ZFNs) and transcription activator-like effector nucleases (TALENs) required a complex and time-consuming process to design and construct custom proteins for each target gene. This limited their applicability and made them less accessible for researchers. Point 2. High cost. The complexity and customizability of earlier gene editing tools made them expensive to develop and use. This cost factor restricted their accessibility to a limited number of well-funded research labs and institutions. Point 3. Off-target effects. Previous gene editing tools often lacked specificity, leading to unintended modifications at off-target sites in the genome. This lack of precision raised concerns about the potential risks and unintended consequences of genetic manipulations. Point 4. Low efficiency. The efficiency of earlier gene editing tools in introducing desired modifications into the genome was relatively low. This limited their effectiveness, especially when working with complex organisms or large-scale genetic modifications. Point 5. Technical expertise. Previous gene editing techniques required specialized technical expertise, both in designing the tools and executing the experiments. This limited their adoption by researchers and hindered their widespread use in various fields. Point 6. Scalability and flexibility. The previous tools were limited in their scalability and ease of use for multiplexed editing or large-scale genetic modifications. This restricted their potential applications in areas such as therapeutics, agriculture, and synthetic biology. The development of CRISPR-Cas9 gene editing technology has addressed many of these limitations, offering a simpler, more efficient, and highly versatile system for precise genome editing.